Well, thank you so much for the invitation. And I see there's a lot of discussion already ongoing in the chat. So I'm happy to also answer some questions later. Um, I am a physician scientist, originally from Germany, now working in the Netherlands, focusing on different types of genetic therapies. I already got quite a few questions before this meeting started via email asking me to include patients into trials for genetic therapies. And I just want to say that right now there's no trials ongoing. So we don't have any genetic therapies at the moment. I just want to give you a bit of an overview of what is possible generally for the genetic therapies, but also discuss some of the limitations of what we can do at this moment. And I kind of want to repeat a few things that we've already heard about just to have the information again. And I think Emma wonderfully explained, even though the CLCN4 related condition is considered rare, there are so many rare diseases worldwide that 6 to 8% of the population are affected. And we're talking about millions of patients across the globe. And 70 to 80% of them are genetic in origin. That means we can find the cause of the disease in the DNA. And I think you know that for CLCN4, but for many other rare conditions, we do not yet have any targeted therapies available. That means we are relying on symptom management and really case by case trying to figure out what is working for the patients. And what we eventually want to have, and Emma mentioned this as well, we kind of want to try to identify what works best for each patient and then really on a genetic level try to treat. Do want to say most of these treatments won't be a cure, but they can have a significant benefit on the patient and their further development. And there's many different types of therapies out there that we consider genetic. And I try to talk a little bit through them and tell you what is possible and what is not. And I want to go back into like basic biology. We talked about the DNA, so the genetic information that basically makes us who we are. And Emma said we have lots of differences on the DNA, showing our height, eye color, whatever. And the DNA is transcribed in what we called the messenger RNA. And this is really the message that goes out that helps the cell make a protein. And then from the mRNA, which goes out of the nucleus, so in the nucleus, the DNA is protected, it goes out of the nucleus, and then we produce these proteins, the cell makes the proteins, and the proteins then have specific function, like CLCN4, which works in the cell membrane with the ion channels. And along this axis, there's many types of interventions we can do, and they have different effects. So on the DNA level, we can do a gene replacement. So we can introduce a gene that is not functioning. We can do something called genetic engineering. That means we can alter the DNA that is already in the cell. And both of these therapies are permanent. So once you put them into the cell and you alter something on the DNA level, it will most likely be permanent. On the RNA level, we can do so-called RNA therapies. And I'll talk about this in a few minutes. And the RNA therapies, we call them transient. So they have a limited effect along the time. And that's because we continuously produce new RNA. And then we also degrade the RNA so that the RNA goes away. So any effects that we have on the RNA that will last for a few days, weeks, sometimes even up to 6 months. The advantage here, even though you have to reintroduce the drug multiple times, if you have any severe side effects, you can stop the drug and the side effects might go away. And the last thing we have is what we call enzyme replacement therapies. So instead of the developments on the DNA or RNA level, you can just try to give the protein back into the cell. Also, these effects are limited in their duration. Normally, they last 2 to 4 weeks, so very short. And depending now on what is possible for a specific condition and what we need, we can choose which is the best therapy. And the best therapy also depends on the inheritance. And I think you already heard this just a few minutes ago. There's different types of inheritances. There's the one that's basic for the chromosomes 1 to 23, which we call autosomal inheritance. And we have the dominant inheritance and the recessive inheritance. We have two copies of each gene, and if there's a dominant inheritance, one copy of the gene is affected and someone has a disease. And if you have a recessive inheritance, you have to have both copies affected to develop the disease. That's what we see with chromosomes 1 to 23. But then we have the sex chromosomes, so X and Y, and CLCN4 related condition is a sex chromosome related condition, so X-linked. And we already heard in females, one of the chromosomes has to be affected, and then the females can develop the disorder. And for the males, they only have one of the X chromosomes. So if that is affected, then they will develop the disease. So once we know the inheritance pattern of a condition, we can already start making some decisions on what would be a suitable therapy. But the other thing we also need to know, what is the functional effect of one of these genetic variants? And I think we already heard that right now. I just want to repeat it again. And I'm showing this on a very simple example. So this is not CLCN4. This is really just an imaginary example. So this is an enzyme that has a black rectangle, and it changes it into a gray triangle. And if we have a loss of function, that means that it doesn't work anymore, and you can have it now affecting two of the genes, the copies of the gene, or only one. So this one is only one. So the yellow one inherited from one of the parents doesn't work, and the purple one inherited from the other parent, that is still working. So half of this is not working anymore, but then you have the disease, and it's a loss of function. What you can also have is what we call a gain of function. So it's overworking, it's producing too much. And if that's happening, we call it a toxic gain of function, and that can also cause a disease. It's just overfunctioning, so everything in the cell is getting out of order. So that's what you can see here. And then we have something that is quite complicated, but it happens in multiple diseases, and that we call a dominant negative effect. Sometimes proteins work in pairs or couples or multiples. In this case, I just made them in pairs. So if two of the healthy copies go together, that's fine. They're working well. If two of the unhealthy copies go together, nothing is working. But now we can also have the condition where one of the healthy and one of the unhealthy copies go together, and then the unhealthy copy makes the healthy copy also not work. And we call this a dominant negative effect. So it kind of is like a domino effect. It takes the healthy copy down as well. And these are actually very severe cases where you have little function left overall, because the healthy copy is also affected by the unhealthy copy. So once we know what's the inheritance pattern and what is the pathomechanism of the certain variant, we can start making decisions on what is the best therapy. And one that is very easy is the gene replacement therapy, and then works with loss of function variants. So I either have one copy lost or maybe both copies lost in autosomal recessive diseases. And what I can try to do, I can replace the defective gene. I'm actually not replacing it. I'm adding a healthy new one. I'm not taking the unhealthy out. So everything stays in the cell, but I'm adding a new healthy copy of the gene. I'm introducing it to the cells. And that is often enough to restore the cellular function. That is one thing we can do. Gene replacement therapies have to be delivered with certain type of vectors, as we call them, and mostly they are modified viruses. So they're not infectious anymore, and they don't do, they're not pathogenic for the cells anymore. They just introduce this new copy, and that works well for brain disorders. However, it doesn't yet work well, for example, diseases of the gastrointestinal system. So you could maybe treat the neurodevelopmental effects or the seizures, but you would not be able to treat the gastrointestinal problems. So this is something we always have to consider with all of these therapies. Another thing we can do on the DNA level, which I discussed, was the gene editing. So we can have the DNA where there's some defect on the DNA, and we can use a molecule that is called CRISPR. And I think CRISPR has been all over the news in the last few years, and they call it like a scissor that can just cut the DNA. So you can cut the DNA, and then there's different types of CRISPR molecules, and they can help replace this part of the DNA that's not functioning, that has this mutation or the pathogenic variant, and then kind of replace this and correct it on the DNA level. And in this sense, it is absolutely independent what the variant is and like what the consequences, like loss of function, gain of function, dominant, negative, because you're really correcting back to the normal copy of the gene, so to say. Gene editing at the moment, unfortunately, also has to be delivered with certain type of vectors. And best at the moment, it goes into the liver. That's the best organ we can target right now. We can also target the blood. We still have problems going into the brain with these type of vectors at this moment. So this is a limitation. We hope to overcome this in the future. Then we have the RNA therapies. And there's many things you can do with RNA therapies. And I'm just explaining two of them. If you have an area of the RNA that has a mutation and a pathogenic variant, there's these molecules that we call antisense oligonucleotides. You don't have to know how they explicitly work, but they can help you take out parts of the RNA. And then you have a truncated protein. So you lose a bit of the protein, but it can still be functioning. And that you can use for any type of the variants, like loss of function, gain of function, dominant, negative. The other thing what you can do with these RNA therapies, if you have one of these hyper-functioning or misfunctioning mutations that are gain of function or dominant negative, you can knock down that part. So you can degrade the mRNA, you can get rid of this unhealthy copy of this mutated copy and then only have the healthy copy left. And that can help you a lot with many diseases, and there's quite a lot of these treatments out there already. And these antisense oligonucleotides, they also can very well go into the brain, for example, and can treat neurological disorders. Just to give you an idea, like what would this look like? So here you have the unhealthy copy and the healthy copy for this one of the gain of function variants. So this is overfunctioning, and you can use the antisense oligonucleotide. It will degrade the RNA and only the healthy copies left, or sometimes you don't have it 100%. So some of the unhealthy copies still left, but you have way less. And this way you can, for example, correct a gain of function variant. So these are things you can do at the moment, but all of them are limited to certain tissues, certain type of diseases. So to just summarize, the therapeutic strategy really depends on the genetic variant, the inheritance pattern, and the mechanism of disease. And I think you just heard from Michael, there's different types of mechanisms we know for CLCN4. So you have to pick what is best for each variant. We have still very big challenges to deliver the therapy to different target tissues and organs. So depending on the variant and depending on the delivery method, it might be possible to go into the brain, but might not be possible to, for example, go into the gut. And I think this is something we have to discuss as well. The developments are very time consuming, so they can take multiple years, and they're very costly. So an antisense oligonucleotide for a single patient can cost up to $1 million. And a gene therapy or a gene editing therapy for a single patient can cost $2 million. So this is really high costs. So what we're trying to do for different types of diseases, we are trying to identify if with one therapy we can maybe group patients so that we don't only treat a single patient, but we can at least treat like 10-15% of the patients so that we can distribute the costs over multiple patients and not have to develop for every single patient themselves, because then it will take too much time at the moment and it will be too costly. We hope it will get cheaper in the future, but this is something I just wanted to mention here as well. And with that, I'll stop and give back.